My name is Rinbok Wi. And I'm the downstream tech transfer at Samsung Biologics SMEs. Today, I will explain the end-to-end technology transfer process, including practical examples of Samsung Biologics achieved successful drug substance technology transfer. This webinar is Technology Transfer 101, which provides an introductory basic knowledge regarding the technology transfer. Today's webinar deals with the introduction of technology transfer and its role especially for the biopharmaceutical industry. End-to-end technology transfer process will be explained, and I will also share this technology transfer at Samsung Biologics. Okay, let's begin with the introduction of the technology transfer. What is the technology transfer in pharmaceutical industry? ICH Q10 Pharmaceutical Quality System defines the technology transfer as the systematic process of transferring product and process knowledge between development and manufacturing, and also within or between manufacturing sites to achieve successful product realization. Each product has four life cycle stages, such as pharmaceutical development, technology transfer, commercial manufacturing, and product discontinuation. Technology transfer is the first stage of GMP activity. Knowledge on manufacturing process, control strategy, process validation, and continuous improvement enables GMP manufacturing and commercialization of the product. Now we know what is the definition of technology transfer. Before we move to the technology transfer process, it is good to see the role of technology transfer in pharmaceutical industry. Why is the technology transfer important in drug substance manufacturing? Technology transfer is a critical component ensuring manufacturing readiness, product consistency, and regulatory compliance. It is essential for mitigating the risk and accelerating time to market through effective transfer process. A robust and standardized approach to the technology transfer ensures continuity from development to commercial supply while maintaining product quality and regulatory integrity. CDMOs can provide benefits for the accelerate product launch through effective technology transfer. Drug substance developers have common challenges such as limited manufacturing capacity, lack of resources to launch product commercially, and lack of marketing and distribution capability. CDMOs can support these challenges. With CDMO, it is possible to accelerate pharmaceutical product launch without delay and cost incurred by having to build and validate a new facility. Experienced technology transfer SMEs from CDMO shorten the time for process knowledge transfer and process validation for earlier commercialization of the product. In addition, CDMOs typically have dual sourcing strategies to mitigate the supply chain risk, which enable fast and continuous product launch and supply. From the first two sections, we have reviewed the introduction and role of the technology transfer in pharmaceutical industry. And the third section is the main section of this webinar that explains the key stage of technology transfer process. This slide shows an overview of the technology transfer life cycle. Here, I briefly introduce each stage, and details will be further explained in the next few slides. Technology transfer activities start with project initiation for goal setting and establishing a technology transfer team. Next step is project planning. Based on the shared knowledge, plans for manufacturing scale, engineering run, and a small-scale verification runs are established. Execution of engineering batch production and manufacturing scale is the next stage. Operational readiness at the receiving unit GMP site is checked prior to the formal validation campaign, and any risks to be reserved for commercial manufacturing are identified. After all mitigation actions from transfer execution stage are completed, validation campaign is executed, and technology transfer lifecycle continues through regulatory filing and pre-approval inspection preparation to support regulatory approval of the product. Once the product is approved by the regulatory agencies, the product is ready for commercial manufacturing. The first stage is a project initiation stage. The project initiation stage defines the scope and strategy of the technology transfer between sending unit and receiving unit. Team charter, roles, and responsibilities are established according to the scope. And one team can focus on the compatibility of the product quality, but another team can focus on the improvement of productivity and product quality. Technology transfer team will be established and communication channel will be setting to meet this goal and scope of the technology transfer. Communication hierarchy is aligned between the teams for effective communication. One core team for cross-functional discussion and multiple sub-teams for in-depth discussion for each function are commonly utilized for the technology transfer. A face-to-face kickoff meeting is one of the main activities for this stage to address and share key project goals, assumption, activities, deliverables, project timeline, and key milestone expectations. Identifying high-level gaps on the raw materials and process is required before the kick-off meeting or to discuss the risk and mitigation action during the meeting so that the team could start the mitigation action earlier that could affect the overall technology transfer timeline. Through this stage, exchange of comprehensive technical reports that detail the process and analytical methods are studied. The next stage is the project planning. Technology transfer document packages are delivered from sending unit to receiving unit. The examples are shown in this table for your reference. Documents regarding the raw material and active pharmaceutical ingredients, manufacturing process, packaging process, and other supporting data can be provided. Team members from the receiving unit digest the knowledge and process information. It is important that the team not make full assumptions that would otherwise be identified later during engineering run or validation runs with serious consequences to the project success and timelines. Receiving units should request a documented process condition as sending unit to minimize gaps and risk during this stage. Process and material gaps are identified and listed, and relevant risk mitigation actions are required. Some risk mitigation actions like introduction of new equipment or new materials require extended period to resolve them. Therefore, fast identification and decisions shall be made, not to delay over timeline. Based on this gap assessment and facility fit assessment, mass balance is generated and bill of materials are documented to place an order for GMP manufacturing. At the same time, activities for small-scale verification run to assess process compatibility are planned. Similar to the manufacturing scale, gaps are identified and resolved, and mass balance and bill of materials are generated. The next stage is project execution. We prepare the receiving site for process execution through engineering and verification runs. Prior to the validation runs, operational readiness at the receiving site is verified through engineering run. Automation, wet testing, and engineering runs are executed prior to the validation campaign to ensure that the process performs as expected. This stage is the most intensive phase of the technology transfer. Through this stage, all issues identified have been adequately addressed before proceeding to validation campaign. Verification runs and satellite runs as scaled-down laboratory and or pilot models also can be executed to ensure robustness and consistency of the process. The key idea to execute the technology transfer is scale-up. Scale-up is defined as the process of increasing batch size. Reliable scale-up of unit operation is a key factor ensuring the success of a process operation. Process parameters can be classified as scalable parameters and scale-independent parameters. Scale-independent parameters shall be remain the same, but scalable parameters shall be increased or decreased depending on the scale. Here are some examples of scale-independent parameters for typical downstream process: chromatography, filtration, and UFDF. Chromatography resins shall remain same, and bed height, linear velocity, residence time, which relate to the process conditions, are also remain same. But the flow rate, column volume, and the resin volume will be changed according to the scale of the process. For the filtration, filter type and sequence of operation, temperature, flux, and flush volume per area will remain same, and the flow rate will be changed according to the scale of the process. For scale-down model, qualification and execution is required and performed in the similar manner. After completion of mitigation actions from engineering run, the next stage is process validation. Process validation strategy and required supporting studies should be aligned between sending unit and receiving unit, and it is essential that technology transfer execution should consider process validation. An earlier discussion for the validation strategy is recommended as there may be differences in validation strategy between the sending unit and receiving units. Process validation master plan and process validation study protocols are generated and executed during the process validation campaign. Process data are actively monitored and verified so that issues can be identified and resolved rapidly without affecting the overall validation campaign. After completion of process validation, the result of process validation study will be consolidated into process validation reports, and stage will be ready for regulatory filing and PAI readiness. CMC sections of regulatory dossier are generated and submitted to the agencies. Regulatory agency can ask additional data or question during the review of dossier. Both sending unit and receiving unit are responsible to respond to those requests. Regulatory questions typically need to be answered within a specific time, so clear communication plan between technology transfer team is highly required. Pre-approval inspection is a part of regulatory approval as one team mind or relevant technology transfer team members prepare the pre-approval inspection or technology transfer and process validation, including the issues and their resolution, need to be ready for presentation. Once the product is approved by a regulatory agency, the product is moved to commercialization stage. Statistical process monitoring and continuous improvement are key indication for this stage. Process monitoring data will be reviewed and how the team will respond to SPC signal to take appropriate corrective action. For this purpose, TT team, technology transfer team will be restructured to support continuous improvement effectively. This slide shows the examples and key deliverables of technology transfer for each stage. Deliverables from a stage can be a prerequisite for the next stage, and therefore it is essential to document outputs of each stage on time to support traceability, alignment, and regulatory compliance. Okay, if we review the stages of technology transfer, and from this section, I will share some examples of technology transfer activities, what we are currently doing at Samsung Biologics. As a CDMO company, Samsung Biologics supports scale up and scale down with flexible, standardized infrastructure. Sending units of our technology transfer can be client's development side, client's manufacturing side, or Samsung Biologics development team known as CDO. The process knowledge from those standing units are transferred to Samsung's DS manufacturing plant, which have 1,000 to 15,000 liter scale production bioreactors. For Plant 3, Plant 4, and Plant 5, we have dual purification suite, which can further accelerate the delivery of the product. And our MCell Lab is capable to execute the 2-liter and 10-liter bioreactors. And we also have a pilot lab can execute the 200-liter pilot models for process robustness and consistency. Process and materials are standardized to minimize the risk and to ensure the on-time delivery when we perform the technology transfer. Samsung Biologics provide streamlining transfer through consistent materials and process design. Samsung Biologics has the technology transfer guide document, which is aligned with regulatory requirement and our industrial standard in order to maintain the quality of technology transfer deliverables. We also have a individual's career development plan to train the individuals to have the same purpose. The slide shows the overview of technology transfer activities and deliverables at Samsung Biologics. Documentation deliverables are generated on time with our experienced SMEs to ensure the on-time delivery of the product. Samsung Biologics standardizes the process and materials as a mitigation action for on-time delivery of the multiple products. Process conditions are dependent on the development of each product, so they could have a large variation, and this variation of complexity could reduce the efficiency of technology transfer and could lead to increase the human errors. Therefore, Samsung Biologics standardizes the process control strategy and the list of control parameters as much as possible. It minimizes the process variation between products and reduces the documentation timeline and the human errors. In addition, the standardization also reduces the barriers for internal technology transfer, which can support the client forecast. We also use standard raw materials and consumables. It enables fast and cost-effective procurement by minimizing the supply chain complexity. Each materials are also dual sourced to minimize the supply chain risk. Furthermore, Samsung Biologics manufacturing sites have also standardized facility design. This is essential for multi-site transfer by removing variability in equipment, automation, environmental conditions, raw material sourcing, process change due to facility fit, analytical method, or QC testing. Samsung Biologics is ready for internal technology transfer by the standardized facility design, and also we can provide nested PPQ approach, which is a structured strategy where a subset of sites represents the entire network. Through this approach, our client will have benefits like reduced batch requirements, fast market launch, regulatory flexibility, and consistent assurance. Here, I also share some digital tools we are using for technology transfer. The first example is facility fit assessment using mass balance with Monte Carlo simulation using Oracle Crystal Ball software. Typical mass balance use minimum, maximum, and average values of each process parameters, but is highly dependent on the representativeness of the provided data. Monte Carlo simulation can reduce this uncertainty by the representativeness, and also it can provide estimation of a combination of multiple parameters with visualization. The second example is multivariate data analysis model-based monitoring and prediction. Our plan has the capability to perform this MBDA model-based monitoring and prediction. Any unexpected behaviors can be detected earlier and their impact on the situation can be predicted. Online trending and monitoring are available for Samsung Biologics manufacturing scale plan using Synca Online. And 200 Liter Pilot Lab also has the capability for NVIDIA modeling, monitoring, and prediction with Synca. The third example is the Samsung Biologics internet system. We launched a data sharing platform called Samsung Client Portal to collect all the manufacturing data, the quality records, and QC testing results, and also the raw material information into our electronic data warehouse. And the results can be shared to the client with the raw data and chart type. So through this, Samsung biologists are using these tools to enhance decision making and risk management. Okay, that's all I have prepared, and now I will wrap up today's webinar. Technology transfer plays a central role in establishing commercial manufacturing readiness and ensuring process continuity across sites. A structured data-driven approach is essential to achieve consistent and compliant outcomes through standardized infrastructure in integrated digital system. Samsung Biologics supports efficient scale-up transport across a range of biologic modalities. Today, I briefly introduced technology transfer of drug substance manufacturing at Samsung Biologics. We also offer other modalities like drug product, mRNA drugs, and antibody drug conjugate. I hope we can have another chance to introduce the capability of these new modalities in your future. I will wrap up today's webinar with this picture. All members in technology transfer teams are relay learners sharing a common goal to launch the product successfully and rapidly. Thank you again to join today's webinar.
All right, first question. How do you evaluate if a facility and its equipment are fully compatible with the process requirements?
Assessing facility fit is critical, and the capability to manufacture the product needs to be determined through gap assessment. Based on the process data provided from the sanding unit, mass balance is generated. Process conditions, including the batch volume, operational parameter range, and processing time, are compared to the equipment capacity at the receiving site. If the process does not fit to the current facility or require a longer process time, causing delay in overall manufacturing schedule, process modification or introduction of new equipment shall be considered.
Okay, thank you. Next question. You mentioned that the process validation should be considered during the technology transfer execution. Can you please share more details?
Okay. Process validation and regulatory filing are critical components in the technology transfer of biological drug substance for the commercial manufacturing. As you know, it is important to identify the process validation requirements and regulatory requirements clearly from the early stage of technology transfer. A sending unit typically does not provide filing document at the early stage, so there may be a gap between the filing requirements and the transfer data. Therefore, it is important to ensure that all parameters required for the filing have been accurately transferred at the beginning and early stage of the technology transfer.
Thank you. Next question. What is the most bottlenecking step to meet the production schedule during your technology transfer experience? And what was the action for debottlenecking?
Okay. Managing and resolving long lead procurement is critical to minimizing project delays and supply chain disruption, especially in pharmaceutical manufacturing. Our identification of long lead items and incorporate procurement activities into the project plan at the earlier stage. Generating material and mass balance is also important to order the required amount of the long lead materials. We standardize single-use materials to simplify the supply chain complexity and fast and cost-effective procurement for each scale of the manufacturing process.